Welcome to Let's Talk Micro. Hello, dear listeners. Welcome to another episode of Let's Talk Micro. As always, I hope you had a great week. And you can always find Let's Talk Micro on Apple Podcasts, Spotify, Amazon Music, Goodpods. Wherever you listen to podcasts, you can find Let's Talk Micro. As for our social media, I am on Instagram, TikTok, and YouTube as Let's Talk Micro, on LinkedIn as Luis Plaza, and on X as Let's Talk Micro 1. I also have an email address which is letstalkmicro@outlook.com. So either via social media or via email you can send any feedback, any topic suggestions. Those are always welcome and appreciated. So please go ahead and subscribe to the podcast, download episodes, and if the app allows you to do so, please go ahead and leave a review. Thank you so much for the support. As you know, learning about antibiotics is definitely a lot of information, so if you're looking to learn more about antibiotics, please check out www.learnantibiotics.com and the Learn Antibiotics book, available on Amazon. These resources include cheat sheets, practice tests, games, and more, and they are being used by thousands of people worldwide and may be helpful for you or your colleagues. So definitely check out these great resources from Dr. Timothy Gauthier, a very passionate pharmacist. As usual, I always talk a little bit about the previous episode, so in this case it's two of them. As you know, I'm just going to take a minute to talk about this. On Thursdays, we normally publish a regular episode, so if you haven't checked the one from last Thursday, that episode, in it, Grace Lubert, she's the director of the Medical Laboratory Sciences program at the University of Alaska in Anchorage. She joined the podcast and she was in a previous episode where she talked about her program. In this one, she came in to talk about the research that the MLS students do, which is very atypical, I will say, from your regular MLS programs. You normally work on maybe some sort of project, but this is very detailed research. And of course, she goes over the whole process and the results that they have seen, but the students, they collect samples, they collect scat from moose, bear, and geese, and then they freeze them through the whole procedure, then they thaw them out, and they go ahead and plate them, and they screen and they perform testing, and she talks about the organisms that they have seen, including ESBL, producing organisms. So I think this is very fascinating, and the students come out learning so much, especially about antimicrobials, because they do susceptibility testing on those isolates. So, and as you know, learning about antibiotics, it's so much information, and when we go through a program, we learn some, but we barely touch the surface. So it is great that the students come out learning so much about antimicrobials. So shout out to Grace. Thank you so much for what you're doing. And then the latest episode was a bonus episode that came out this past Tuesday, and it was about what I liked in the American Society for Microbiology event, Microbe 2024. Which took place in Atlanta. And, I've been going since 2023, and both times, have recorded a live episode. And basically what I do is, talk about my experience, which talks have I been to. I normally attend podcast recordings. I talk about the posters that I like. So it was a great summary, so I hope you check it out. And if you have the opportunity to attend Microbe, which is the largest event from the American Society for Microbiology and one of the largest events of microbiology, if not the largest. It's a gathering of and people from all over the world attend. So I definitely invite you to attend this event if you haven't already. So I think, if you're a hardcore microbiologist and you're so into it, you will definitely enjoy it. And of course, one thing that I always love when I go to this type of events is just when I meet people that actually listen to the podcast or they tell me that it has helped them somehow. So I definitely enjoy that, just to know that I'm here, week after week talking about a topic and then people are listening. And it is so great when I actually meet those people. So it's a great feeling, and I appreciate so much your support. So I want to do a couple shout outs. Shout out to Alexis, who I briefly met before on 2023, but then this time, I had a chance to talk more. Shout out, thank you for listening. And then shout out to Jessica. Jessica is a fellow MLS, and I had a chance to talk to her for a few minutes, and she listens to the podcast. So Jessica, it was so great meeting you, and thank you for listening. Okay, so let's go ahead and jump to today's episode. So today's episode is about the microbiome, and this is something that maybe we see, we might see some publications and we might not be too familiar with it. We definitely hear terms like microbiome, microbiota, and we see published articles out there about how maybe an imbalance of the microbiota and the composition, it can sometimes, it can be correlated with diseases, and we know that as microbiologists, in different areas of the body, the composition of organisms, it varies. So in this episode, Dr. Crystal Icenhour, she joins the podcast and, to talk about the microbiome. She gives some definitions. Specifically, she talks about the human microbiome, what kind of organisms we typically see, based on different areas, how you can sequence the microbiome, what are some of the applications, and something, she talks about something that's very important, which is, once we do the sequencing and we have the information of the organisms that we have, what do we do with that information, right? As you know, some organisms, are not supposed to be in the body. They are not normal, or sometimes, we have some that are normal in some areas, or maybe you have an abnormal concentration. So I definitely think this was a great episode and a great way to introduce ourselves to the microbiome and those type of terms, organisms and things like that. So it was a great episode, very informative. So I hope you like it. Let's go ahead and listen to it. So those of you that are medical laboratory scientists and microbiologists and you work on the bench, a lot of times, you definitely, your organisms, and you know that some organisms, they're part of the flora of certain areas, and you get quite knowledgeable, knowing, typically what's a pathogen versus what's normal flora. But this is a very interesting topic, and I was in a conference listening to a speaker talk about the microbiome and the microbiota, and sometimes, there's so much information that we might not be too familiar with it. And to be honest, until I started doing graduate study, that's when I started getting more familiar with it. So a very fascinating topic. So I invited her to speak here and she accepted. So I'm very happy about that. So with us today, we have Dr. Crystal Icenhour. Dr. Icenhour, welcome to Let's Talk Micro.
Thank you. It's a pleasure to be here.
My pleasure. So maybe let's start with a quick introduction about what you do and anything else that you want to add.
Sure. So my career's primarily been focused on biotechnology companies. I've co-founded and led two different biotech companies through to acquisition in my career so far. And currently, I'm working for the last acquirer of my former company, LabCorp. So there, I serve as the director for microbiome and specialty infectious diseases.
Thank you again, and we're definitely in the laboratory community. We know we're definitely familiar with LabCorp and definitely a big reference lab. So let's start with some basics here. So, what is the microbiome?
So, well, it's not a singular thing. There are many different kinds of microbiomes. There's a microbiome on the desk, on the floor, in your nose, in your belly button. Each environment has its own unique microbiome. And the microbiome, it's the collection of microorganisms, their metabolic byproducts. It includes bacteria, fungi, parasites, viruses. So it's the whole microbial cosmos that exists in different environments. And each one of those environments, again, is unique. And the microbiome of the nasopharyngeal space is completely different than the microbiome of the intestinal tract. And you can actually tell where the sample originated from by the types of microorganisms that are present in the different spaces.
Definitely, and that's something as we when we work with cultures, we know based on the source, right? So we're expecting sometimes, we see some organisms. Okay, here you can expect to see this versus another area where you shouldn't see. So depending on the sample and the source, it's definitely very varied. So maybe I know, so definitely like it changes as you said, composition like from the oropharynx, the intestines, the genital area. Maybe can you give some examples of the organisms that we can find for the audience?
Sure. So when you're looking at like the oral cavity, for example, because it's one of the most diverse microbiomes that is present in the human condition, and my information will be primarily focused on the microbiomes of the human body, and so the oral cavity has hundreds and hundreds of different kinds of bacteria normally found there. It's very diverse, lots of different species of bacteria. Things like porphyromonas, which has been linked to gum disease and other things, but also a lot of other bacteria, too many to list. When you look at, say, a fecal sample, again, hundreds of different kinds of bacteria, but slightly less diverse than the oral cavity, interestingly, as far as types of microorganisms. Um, and then there you're looking at things like Fasciolobacterium, Bacteroides, things of that nature. And then, looking at something like the vaginal cavity, there you have very low diversity. You're typically looking at 1 to a dozen unique microorganisms present in the vaginal space instead of hundreds. And you're looking at things like Gardnerella and Lactobacillus as kind of the most common things that are found there. So very unique in terms of the types of microorganisms, the species of microorganisms, but also the diversity and the microbial burden. Obviously, a fecal sample has a lot of microbial content. Upwards of 80% of the sample can be comprised of the actual microorganisms. In a nasopharyngeal swab, there's very low microbial burden. So when you do a swab there, most of the genetic information you pull out is actually from the human, the person that was collected from. So lots of different ways of looking at the differences in microbiomes throughout the body.
Yes, definitely composition is it's definitely very varied, and that's something that we see in the lab with the samples, and I remember when we used to do like fecal white blood cells on the stools, like we put them on the slides, they're so concentrated, you see so much bacteria, so it definitely it changes from site to site. So you mentioned, when I was listening to your talk, right, there are some diseases that can be linked to an imbalance in the composition of the microbiota. Can you give some examples?
Sure, sure. The most overt is infection. So if you have a microorganism growing in high quantities in the wrong part of the body, that's the traditional definition of an infection. So that's the kind of the most drastic perturbation of the microbiome that we typically see. But there's a lot of other dysbiosis. It's not an overt infection, and there's nothing there that's acting as a true pathogen. But when you have the wrong proportion of the microbes or the wrong balance of the microbes, you can get dysbiosis, and that is increasingly being linked to a lot of chronic health conditions, things like some forms of diabetes, cardiovascular disease, Alzheimer's, Parkinson's, you know, all kinds of things are being linked to these perturbations of the microbiome. There's even some connections with the gut microbiome and colon cancers and other types of cancers. So yeah, it's really, we're just scratching the surface at understanding, are these dysbiotic states, are they causing the condition or are they a result of the condition? So a lot of very interesting work going on and a lot of new discoveries to be found.
So I know that, like I have a couple questions here that are definitely very heavy on the topic. So I know that if as general as you can, or you can, give as much information as you can, but I know that they're heavy in information. So as far as what are some microbiome applications? Can you give us some examples?
Sure. So my former company, we were actually using microbiome characterizations to help solve medical issues. So patients who were exhibiting signs of infection, but all the traditional tests were coming back negative or indeterminate. So you can, look at the microbiome and see if there's something infectious there. So an example might be, um, you know, you say you have a urinary tract sample and there is, um, high levels of DNA virus or like something like a BK virus, JC virus. They're a normal part of the urinary tract system, but they can grow and proliferate at high levels and cause issues within the body. So things like that are things you can see with looking at the microbiome, things that shouldn't be in the sample type you're looking at. Um, and that's probably the one that's most clinically relevant right now because we understand, no, we shouldn't have tuberculosis circulating in our blood. That's not something we want to see in any healthy individual. There's no normal level of tuberculosis to be found in the human condition. So that's the application that right now has the most direct clinical application and an area that I've worked directly in for a number of years. There's certainly lots of possibilities. When you look at, disruption in the gut microbiome, there's this gut-brain axis that people talk a lot about. And you can see there's disruptions in the gut microbiome, and you can link it to or correlate it to systemic disease, but it's there's not any clinically validated bodies of work yet being able to use the microbiome characterization to be able to help diagnose those types of more chronic systemic issues. I do believe that it will happen, but we need a lot more data. We need a lot more information, and we need and so that side of microbiome is really more on the discovery side. So discovering these links and connections between the microbes and these chronic conditions that we suffer from.
Well, thank you for that. And so what about some methods used for sequencing? Maybe you can talk a little bit about that.
Sure. And there's a couple of different ways that you can characterize a microbiome. All of them require sequencing. So next generation sequencing is the most commonly used. You can, starting with the more simple methods, a lot of groups use 16S sequencing, where they're looking at the 16S gene or a portion of the 16S gene that's found in all bacteria and archaea. So you can, do basically PCR amplification of that gene or fragments of that gene, and then put it through next generation sequencing to differentiate between the different bacteria. It's limited to only bacteria because of that nature. You can do something similar with ITS or 18S genes and be able to see fungal targets as well. But again, there you're limited to just the fungi. So you're missing parasites, you're missing viruses when you have these approaches. But it depends on the question you're asking. And sometimes you really just want to focus on the bacteria that are present in a sample. So it's fairly straightforward. There's a lot of commercial products out there, commercial kits you can use to do 16S sequencing. Mostly it's in the 300 base pair range is what a lot of groups are using as far as amplicon length. One of the other limitations is sometimes because you're only looking at a small fragment of the genetic information for that microbe, you sometimes don't have good resolution to be able to differentiate down to the species or strain level. There's some newer technologies that are looking at complete 16S sequences, and those have much better resolution as far as getting down to species and strain level. But those are not as commonly in use right now. You can also do kind of a targeted approach where you want to pull out just the viruses, or you want to target, the specific populations of bacteria. So, and that's a targeted sequencing approach where you have probes that you use to amplify genetic regions that you know exist in the sample and that you're looking for. And so and that can be useful, like say if you're looking for certain kinds of cancers or things of that nature, that's where it's primarily been used. Or if you have specific microbes that you're only interested in looking at specific microbes, you can use these targeted approaches. One of the more comprehensive methods for doing sequencing is called metagenomic or shotgun sequencing. And this is where you have much more breadth, and it's where a lot of groups are focusing on things. It allows you to identify bacteria, DNA viruses, parasites, fungi. So anything that's DNA based, you can identify with this particular kind of method. And basically what you're doing is it's just like what it sounds, a shotgun approach where you are randomly blasting probes into your DNA sample. And that allows you to get DNA sequence coverage randomly through the sample for theoretically all the microorganisms. And depending on how deep you go and how many sequencing reactions you run, you can sometimes get full length um, genomes for especially smaller things like viruses. But again, it allows you to really take a more broad approach and to be able to see more of the microbes that are present in the sample and not be limited to only bacteria, fungi, or things that you were specifically looking for.
Thank you for that. I know that it's, definitely, and I'm sure the audience appreciates that as well. It's a lot of information, and definitely sequencing is something that we'll hear a lot about, and ultimately a lot of the organisms that we cannot identify in the lab, like we send them and then they end up being identified by sequencing. But we definitely hear some, different terms. So thank you for that clarification. Um, so what is the ketone?
The ketone. So one of the challenges with doing microbial sequencing or microbiome characterization is that there is microbial DNA on almost every surface of the earth, in almost every body of water, on the surface of every human. And so it can be very difficult to get an actual true negative control when you're doing this type of work. Even the molecular grade waters that we use in the lab, there are tiny amounts of microbial DNA present in those reagents. In the enzymes we use to perform the sequencing, to perform the amplification reactions and the sequencing reactions, those enzymes are often created in industrial vats where they're being synthesized by microorganisms of various sorts, usually bacterial, like thermophilic bacteria. That's where we get our DNA polymerases from. And so those enzymes have a not insignificant amount of residual DNA from the microorganisms that they were synthesized within. And so these things carry through your entire process. And so it's almost impossible to get a true negative control running through a sequencing process looking for microbes. So the ketome, and there are a number of publications describing this, there are certain like thermophilic bacteria, water-based bacteria are very commonly found in the reagents we use to sequence. And so really the best strategy is, obviously using great lab practices to prevent contamination or introduction of any microbes from the environmental surface you're working within or from the people that are handling the sequencing reactions. You can UV irradiate things like water and buffers that don't have any enzymes in them to try to cross-link any DNA that may be present in those reagents to try to keep the background as low as possible. But really, you have to run a negative control with every sequencing reaction because you have to know what your quote unquote background is. So the ketome is basically your background level of microbes in a sample. And these, 16S sequencing is very sensitive. Metagenomic sequencing is quite sensitive as well. And so even though you have a negative control, you extract it, you do a DNA quantification, you have the DNA present there is below the limit of detection, but you still put it through to the library prep process. It's still below the limit of detection. You still get microbial sequencing reads in that well. So it's a challenge. So you have to characterize what that, because it can change from depending on what vendors you're using for reagents. If you have unusual environmental conditions in the space you're working in, you really have to do a negative control with every single reaction so that you know what your background is, so that you don't inadvertently include the background microbes on a patient report or um, or interferes with how you analyze your data. So that's a lot of information.
Yeah. Yeah. Thank you for that. So right, so you perform the sequence and then you're looking at the data and so what questions should we ask ourselves, when interpreting the data? I know that you gave a great example, like with an application that if we, seeing something like Mycobacterium tuberculosis, that's something that, it's not normal. So, what questions should we ask ourselves when we're interpreting the data?
So I typically work through kind of three high level logic points. The first is, does this microbe belong in the sample I'm looking at? The example of Mycobacterium, I had a patient that we worked with a number of years ago that presented with arthritis, chronic, arthritis. She was diagnosed with Lyme arthritis, and she had gone through, I think, 2 or 3 rounds of treatment for Lyme disease, and she would get better when she was on the treatment, but then she would relapse as soon as she got off of the antibiotics. So in that particular case, her doctor sent us a blood sample and we processed it through. We primarily focus on metagenomic sequencing. There were, I think, 270 fragments of DNA that aligned exclusively to Mycobacterium tuberculosis in her plasma sample. That's not normal. There's no normal level of Mycobacterium tuberculosis in a human sample. Um, so that's an example where that does not belong, and there's certain things like that, or rickettsia, borrelia. There's a lot of zoonotic microbes that do not belong in the human body at any level. So that's kind of what the first tier is: does this belong in the sample I'm looking at? um, Kind of the second tier down is it in the right part of the body? So E. coli is a perfectly normal part of the intestinal tract, but you don't want to see it in the oral cavity. You don't want to see it in the urinary tract. Those are common causes of infection. So finding a bacteria that is in the wrong part of the body can be another clue on how to interpret that finding. And then the third kind of high level logic point is it in the right amount? So again, taking the E. coli example, E. coli is a normal part of the intestinal tract, but it's typically found at less than 1% in abundance in a healthy gastrointestinal tract. If you have a patient that has E. coli at 30% abundance, that's way too high. So, it's clearly growing, out of proportion with a normal, healthy, microbiome state. So that's an indication of infection. And then all of these things really have to be wrapped up with the um, information about the case, especially most of my work again involves clinical applications. So, the patient history, does this patient have symptoms of a UTI? Do they, do they have, did they go to India or someplace where tuberculosis is, more common? Do they have, history of tick or insect bite? So these things can kind of help build the case where you find something that doesn't is typically not found in a healthy sample, but you kind of build the logic and the case around it on, is this causing the disease the patient's exhibiting?
Yeah. And I know, you definitely, you gave some cases when you were giving your presentation. And I wanted to ask you about, can you talk about the one with the culture negative UTI case that you talked about?
Yes, yeah, we worked with a lot of those with my former company, and what we know about urinary tract infections is um, upwards of 50 to 60% of urine cultures are negative for symptomatic patients. And that's a problem because someone who's symptomatic for a urinary tract infection, that it's very uncomfortable at a minimum, and it can actually be debilitating. So one of the cases that we worked with, there was a patient who had suffered from recurrent UTIs for over 30 years. So, the majority of her adult life had been spent in this state. And so we got a urine sample from the patient, processed it through our metagenomic sequencing testing. And there was, in this case, it was actually interesting. It was 90% of her sample was Bifidobacterium brevi, which is the primary ingredient in a lot of probiotics. But it's not a normal part of the urogenital tract. It's not a normal part of the bladder. And so in this case, it had colonized her bladder and had been causing symptoms of a UTI for a very long time. And then we had other cases. We had a pediatric case that was very similar, where her urine culture was negative, and she was only 3 years old, so she wasn't quite fully verbal yet, in a lot of pain. And unfortunately, her mom found my former company and was able to send us a urine sample, and we identified the bacteria that's not capable of growing in urine culture. And again, the Bifidobacterium brevi from the prior case, it won't grow in a standard urine culture. So you're not going to see it. So in that case, we were able to identify a bacteria that you just would not be able to identify from urine culture alone. So lots and lots of cases for culture negative UTIs, unfortunately.
Wow, that's definitely got to be frustrating when, yeah, especially like the patient, you have symptoms and you go in and there's nothing growing. So, um, yeah, one of the things, that not all bacteria, are cultural, but at the same time, when we talk about urine cultures, we have a more limited setup because of the nature of the organisms that typically are seen in UTIs, where most of them they do grow in your standard, like let's say bullet agar or McConkey. But there are some limitations to that. Whereas when you do other parts of the body, let's say, you add chocolate and other media like anaerobic media, and then you, based on the organism that you typically seen. So, and urines, the setup is more limited. So there's also the potential to not recover some organisms.
Yes, and I just to elaborate on that point, and just bring this back to the patient. There are millions of women that are suffering with culture negative UTIs right now. It's a huge problem, and it's one of the reasons why I get up and I do what I do every day, because I want sequencing type testing to be more commonly available for these cases where people fall into this loop, because they go to their doctor and they do a culture, it's negative, they send them home, they're very resistant to give antibiotics without a positive test. um, and it's you really do need that, because we need to be good stewards of antibiotic usage. But these patients, there's a 7 to 10 year cycle for patients that fall into that to get diagnosed, and a lot of times they end up getting diagnosed with something like um, interstitial cystitis, which is idiopathic. We don't know what causes it. It's inflammation, and so these patients are basically just sent home, told there's nothing wrong with them. It's all in your head. I've had patients who were put in psychiatric wards because they got defiant, because their doctor couldn't give them an answer as to why they were sick, but they knew they were sick. And unfortunately, it spiraled into a "there's nothing wrong with you" conversation. So it gets very complicated, and it's very hard for these patients to navigate through when we don't have the right tools for the doctors to use widely available so that they can get answers.
Yeah, and that's something that all of us can resonate with. That's something very frustrating when we are not feeling well, and we go seek treatment, and then they say, there's nothing wrong. So it's a very frustrating feeling, and not knowing what's wrong with us, what's causing what we're feeling, and it's not very challenging, and it's frustrating. Um, so I know you mentioned that any other cases that you have before I jump to the next question?
There was another interesting case that we worked. It was I think he was in his 30s, had been sick, and had had 2 strokes. Young man, shouldn't be having strokes. No family history of strokes. Um, and I think his odyssey was about 10 years to get a final diagnosis. It turns out and we received a plasma sample from him and ran it through our system. And we found Rickettsia felis, which is a bacteria that's normally found in fleas on cats. Turns out he had a cat. The cat had fleas. So both he and the cat had Rickettsia felis infection from this. But again, it's not something that is one of the standard things that the clinician knows to look for. And that's one of the beauties of using tools like this. You don't have to know what you're looking for. You don't have to do a differential diagnosis. You can just run the sample and see what the data says.
Wow, that one's definitely very interesting. Um, so you mentioned, when I asked you about the applications, and you mentioned some that there's still more information. Anything else that the future might bring? Any future applications that involve the microbiome?
I do see a role for especially gut microbiome testing to be used for preventive care, where you go in for your annual checkup, and one of the standard tests they run is they do a gut microbiome assessment and look for subtle changes that could be there that in the future we can definitively link to Alzheimer's or cardiovascular disease, and then have preventive care offered much earlier than after symptoms start to appear. So that's where I believe the possibility is there to go there, but we need massive data points to be able to make those kinds of connections and correlations.
Well, thank you for that. And as we're coming to the episode is coming to a close, is there anything else that you want to add? Well, once again, this has been, very informative, and I really I enjoy listening to it the first time, and I definitely did now again, and I hope the audience does too. So I just want to thank you again for taking the time to be on Let's Talk Micro.
It's a pleasure. Thanks for having me.
My pleasure. And that, my dear audience, is the end of this episode. I hope you enjoy learning about the microbiome. As always, I enjoy sharing this information with you. Please continue bringing that passion to what you do. It's so important. You do such great work. And thank you so much for the support. Thank you for listening. It's always great meeting you and meeting new people. So thank you so much. And stay tuned. Great things coming your way. So, as always, stay motivated, stay safe, and of course, continue talking micro. Until the next time. Bye.